Okay, let's start the ggplot and data visualization. So from these chapters, we're going to cover how we explore the data and how we think about the values and numbers we get from the experiment and our data set. So the entire chapters from chapter 7 to 12, we're going to devote to learn data visualization. And this part will be cover all the visualization part you're going to do in our analysis and also your programming exercise. So we're going to cover the data visualization and also the introduction of components in data visualization. And some of the tutorials we're going to do with ggplot2 will be covered in an offline class. Okay, so before we moving on to what the data visualization, this is a good reference you might want to have. This is the book by Klaus Wilk, Fundamentals of Data Visualization. So if you click this, and the book is again, this is written in ggbook, so you don't have to buy that. You can just click that and you can go through the introductions and the code practice you can find from here. I found this book is quite useful because again, this is quite latest summary of how we use ggplot2 and also data visualization, but also there are many different the tutorials and exercise you might want to find as an example. And also you can also go to this link. This is the this link. This is the official manual page for the ggplot2 package. Again, ggplot2 is part of Tidyverse ecosystem, only designed for the data visualization. So ggplot2 has a manual page in the philosophy of Tidyverse system. So you can go through from the installation and also the easy examples for your code practice, and also a bit of the example data set as well. So if you click the reference here, then you might find the basics of how you do plotting and what is the layers and different geometries. Like if I want to draw the bar plot or line plot or the box plot or blah blah blah, you can use this as a reference, then you can follow the instructions. So one of the examples is geom point. This is one of the easiest one you might find in your analysis. You can do only 2 lines of code, and you can just follow the ggplot2 meaning. I found ggplot2 official page is quite useful, but also you want to make some, the variety when you do ggplot data visualization, because, you have to opt to your own purpose, and also your experiment has to have some unique features you might want to empathize. So in the case you want to do some custom visualization, you want to do more customization. So that case, I found this page is quite useful. This is called STHDA, Statistical Tool for High Throughput Data Analysis. They contain so many good manuals and blogs about the Tidyverse system and also the R programming. So if you scroll down, then there are some basic things, and you can also find like box plots and violin plots in each example. So if you click the violin plots, then you can get some, the instruction how you do the basic plotting like this. And if you want to add summary stats or mean and median, sort of the customization for the data summary, that case you can also use these functions as well. And also you want to change colors and how you represent and layouts or the legend position, all of each scenarios you might consider during your programming. STHDA contains everything for you. So go to here and read and find the solution you're looking for, right? So why I'm introducing these two pages, the ggplot2 page is obviously the official page, but STHDA is one of the the neatest thing you can find. So I mean, if you want to search some information about ggplot2 in Google, some of the your search might lead to name of log or history, whatever, written in Korean, and those are mostly quite poor and also not very consistent across their explanation. So don't go to the explanation by Korean. I mean, some might be very good, but I kind of find really good things, consistently explaining ggplot situation across the blog post. So go to this page, and this might be the things you might want to have for your entire career. Okay, so that is sort of good references. Let's back to the slide. Okay, so what is the data visualization? So data visualization is basically like finding the story, right? Story of your data set. So these days we call sort of the big data era, or in other words, the era of information overload, right? So we can access to so many data sets, we can generate so many data sets. So if you think about the biological data set, I explained many times, biological data set in 20 years ago, you can just subset the piecemeal information, like a very handful information you get from experiment, right? But this time is totally changed nowadays, right? So let's think about like a molecular cloning. The cloning at a time like an 80s or 90s, you do one cloning, will do like a whole publication. But this time, the cloning by one person per day can be like tens of them or hundreds of them, because the experimental technique has been developed, and also the resources and materials is improved a lot during this time. So that means that you can generate so many data set in these days, right? And also another example is like, when I start my research in graduate school, when I start my PhD, I generate the exome sequencing data set of 200 individuals. At a time, generating the 200 individuals are huge scale for the exome analysis, right? But nowadays, like, my laboratory, the graduate students, like a master's students, they join the lab and they access to like thousands, the exome sequencing individuals or 10,000 individuals, and they analyze the data set. So the scale of the data set is totally changed at this moment. This is because of, the generation technique has been developed, but also the data set you can access from the public collaboration or the research group also increasing the size. So we have to deal with the access of data set and access amount of information, and you have to come up with certain strategy to how you handle, how you filter this information for your story, right? And also raw data becomes useful when we apply method of deriving insight from it. So, I mean, the sentence is quite, complicated. In other words, we can say in this way. So in old days, like in 80s or 90s, when you access to data, so when you're talking about the data means that the final outcome, right? So when you access to the data has to be manipulated, or data was cleaned by someone, and the data is provided in that way. But these days, when you analyze the data or when you do some project, you have very high chance to get a raw data, so data from the machine, right? So why we access to raw data in these days rather than the final outcome? The reason is that final outcome is already, processed a lot, and the sources underlying those data set is already filtered by someone. So you want to access to the core part or other dimensions you might find in a raw data. So you access to the raw data, right? So raw data has many different dimensions you want to explore or you want to parsing. So everyone in these days wants to go to raw data and have it and process in their perspective rather than others' perspective, right? So accessing to raw data in these days is also become, possible, right? So raw data is not kept secretly by, the producers. Raw data is shared by everyone, so you can access to the data set and search that information for your own purpose, right? So this is another way we want to get some data set as well. So one of the examples I can point to is this database. So it's called GEO database. In other words, it's the full name is Gene Expression Omnibus. So if you click the one, you go here, and this is the Gene Expression Omnibus. And what we can do is this GEO database is public functional genomic data repository supporting whatever that's, the alliance. And you get a raw data from this database, right? So which types of raw data we can access? So let's say I want to get to some information about depression. So you search the information about depression here and search, and how many data sets stored in this GEO, right? So you have about 4,000 results for the depression and 350 something thousand for the depression. So they have different levels of layers. So let's look at this, the small one first. So looking at here, so we have 4,000 of data set about the depression. So let's look at the first one. So this is the data set for looking at the sex differences under the stress condition in this mice strain, right? Too slow. Okay. What is the data set for? This is the expression profiling by high throughput sequencing. So this is probably the RNA-seq, and here RNA-seq, yes. And this study data set has the 16 samples. So we get to the 16 samples raw data set from this page, right? So the numbers we see 4,000 and 350,000. Why that is different? Because the 4,000 is the number of studies, and whatever the 300,000 is the samples. And if you go to here, and you can see that the sample, right? The sample description, CBM1. And this CBM1, what is the CBM1? There must be description. Okay, so that's probably here, right? So there are some conditions, right? CBM, CBF, CSM, CSF. So MF probably the male and female, right? So male and female. Is that correct guess? Okay, female, yes. Okay, and what is the B and S? B probably brain, and S probably, what is S? Brain. Oh, no. Okay, let's go here. So CSM again, brain. So PS, not brain and something. Right. So The B is non-stressed. S is the stressed one, chronic stressed one. So this is the conditions of whether mice get stressed or not. Okay, so they have 4 different replicate because, one mice not going to be justifying the result. So they have 4 different mices, right? Okay, so if you go to here, then you click one of the link here, then you can download the raw data from this page, right? So it has the sequence range information for the raw data. So you know about the sequence raw data. Sequence raw data is the FASTA-Q. So if you click one of the link to the data set, then you can find the link to download the FASTA-Q file. Okay, so this is about the data set, right? So this is the FASTA-Q data set you can download from this database. In old days, the data link has posted in here like an FTP something. Then you can click the data, you can click the link and you can download the data set. But these days, the systems are totally changed. So now you have to use the cloud computing. So if you want to access to the raw data set, then you have to go through the cloud computing and you have to know how to use AWS, Amazon Cloud, or the Google Cloud. And this is another layers you might want to learn in future. I probably have to spend some time to explain the introduction of cloud computing. I didn't think at the first time it was essential for biology students, but this has become very universal, right? So I will deal with the cloud computing at least one week in this semester. But if you want to learn more about that, there are some the general class for cloud computing is delivered by the data science the general lectures. So you can find that class, or there are some workshops during the vacation by the data science institute in Korea University, and that was happening last August. Those things might be useful for your learning as well. The cloud computing is inevitable systems you have to learn, not just for bioinformatics, but also for other biological research as well, because all of the computing system going to move to cloud and computing sometime, right? So anyway, there was another thing. Okay, so this is how we access to raw data, and you can also access to the process data set from here. So this is the data set from the RNA sequencing. It's we call the summary statistics. And this data set is processed by these guys, right? So they summarize how they process the data set and how they process the data analysis and all of that information from here. So what I want to make a point in this example is that you are not just access to the summary data set or the process data set, you can also access to the raw data set, because if you look at this description, then you might find that, I wouldn't do processing the RNA sequencing data set in this way. I want to change the strategy. I want to improve the strategy in this way, and I want to get the raw data set. And that purpose, the getting the raw data set is the essential. Or you want to compare this data set with other data set about this depression, then in that case, you can also download the raw data set and comparing to each other, right? So why we have to understand this concept, why we have to get a raw data access philosophy for your biological research. The reason is that, as I mentioned, that the information in these days getting increased, and also in a company or in a research institute, in anyhow in your career, you might be in a situation where you have to get a raw data and compare between the data set and find the candidates and set up the hypothesis for your experiment or product design, right? So this is very common thing. So for instance, like if you work in a drug company, so you're developing the drug, right? And you want to find some molecules acting in a certain drugs or causing some disease. In that case, you want to find the causal genes or causal protein or causal phosphor modification. In that case, you're not just looking at the one data set, you're looking at multiple data set and find the common causal problems across the data set. In the case, you have access to the raw data set and you're comparing the patterns across the studies and hypotheses, right? This is very common practice in a company and also research institute laboratory as well, right? So getting the raw data, you have to get a sense that this is the direction you have to deal with the information, and also how I can find the meaningful information from these complicated dimensions, right? So in the case, you have to learn how to visualize all of that information and how you deliver your findings to others, right? So visualization is such a critical steps you have to do in your data analysis, and getting the very good skills about the data visualization is the key things you can handle those information, right? So that is about the big data aspect. And another reason is that so humans are very visualization driven, creators, right? So we are visual creators. So, when you deliver your information to others, your friends, your peers, your family, your business partners, delivering the lots of text is not useful strategy, because, people easily get a patterns from the graphs or the plots or something you show to people, right? So some of the shapes or colors sometimes is more powerful than just suggesting text to others, right? So in this aspect, the data visualization has to happen for your communication. And again, this is another the notions why you have to learn the visualization, and also it's an efficient way to communication. Okay, so how we can get a data from the data set. So how we can get information from the data set. So in a situation without the the visualization, if you look at the raw data, this looks like the texty, whatever, the two-dimensional situation. So this is the example from our tutorial set, right? So if you're sharing these tables, and we can come up with some questions like, what do we learn from these tables, right? So can you grab information easily from this table? Yes or not, right? And let's think about, if we're delivering some questions like, which states have the smallest population? Is it easy to find from this table? Yes or not? It's obviously no, right? I mean, this is just 6 rows of them. But let's say if you have the 40 rows in this table, then it's not easy to find the smallest value. So in this situation, without the visualization, it's really hard to get a sense in a one glimpse, right? So that's why visualization is often helping the situation where people taking information, people digesting values from your suggestion, right? Okay, so again, the data science workflow, we get a data set and we transform some way in a tidy format. Then we get a visualization and visualization again and visualization again to describing fully about your data set, right? So this is the one way we do that. Okay, so let's move on to how we get information from data visualization. So how we make a sort of good structure of data visualization strategy. So now we are start thinking about, how we suggest colors, shapes, or the data points to others by visualization. So why that is critical. Okay, so let's start off from this two shape, which is quite, the easy things, right? So let's think about the what we can read from these two dots. In a normal situation, like, in offline class, I have to give a question to you, but this is the recorded one. So let's a little bit have some time to pause and think about, what you can think, or you can just press the space and think about that, and I can follow up by explanation. So what we can read from here. So there are two circles, and what we can find from here is these two circles are different size, right? So at least you can find two things from this shape. So first one is, okay, so there are two data types. Oh, sorry, there are two data points, right? So there are two information describing, right? And the second thing is these two information has different quantity, right? We don't know what quantity means, but at least the scales or amount or weight or whatever the high and low values about the data points, they are different, right? So two data points and they have different quantity. And let's move on to next slide. What we can reckon from this information. So compared to previous, it has different colors in a one circle. So what is different? So what we can get, so it's still two information, two data points, right? And they have different quantity and different colors, and the colors representing groups or categories, right? So we can say that these two data points, there are different entries, but these two entries might have different groups or different categories. Like, first year students or second year students, or the A university or B university. So in a previous, let's say it's a previous, the two circles, there are two different entries, like person one and person two, and these two persons from the same year. But let's say here, there are two different people, but the first person and second person, they belong to different groups, like different years, first year or second year students. Okay, so colors here added one more layer. So layer means the dimension, which is the describing another information about the data points, right? So data shape in circles here, they indicating that they belong to the same entries, same groups, but differentiating colors give you different groups, right? So like an year example is here. Okay, so you add one more layers by color. So colors can use in this way. Okay, so we add one more object in this plot. So now we add a arrow under the color, under the circles. So what we can get from this information. Okay, so let's forget about the year of students or something. We just talking about the general situation. So this example here, adding up the arrow, we add up some direction, right? Arrow indicates some direction, right? So it has the start point and end point, and the direction from left to right. And what we can represent by which information we can represent by direction, right? So direction means that literally the locations, like from west side to east side, or direction can mean the time or progress or procedures, right? So it can mean that the early time and late time or something, right? So here again, we add up the arrow in this plot, and we add up one more layer to represent the data, right? So we added the arrow in here. Okay, so next example is what we can read from here, what we can read compared to previous slides. So what changed? So first change is that when we place the circles, we place the blue circles a little bit on the top of the gray circles. So they have different height, right? So what is describing? So we can read that there must be another layers of quantity, right? So we represent the quantity by the size of the shape, size of the circles, but we added one more layers, right? So what is the extra layer is the height, right? So if you look at the vertical layers on here, the gray circles is a little bit lower than the blue circles. So they represent another quantity. So in terms of the circle shape, blue has smaller quantity than gray, but in terms of the vertical layers, it has higher quantity than gray circle. So there are two dimensions to describing the quantities, right? So how many dimensions we get from here? So we get a shape, we get a colors, we get an arrow, and we get a circle size, and we get a vertical height, right? So we have at least 5 dimensions to describing something. Okay, so what we can add more, we add here is a vertical arrow. In other words, we can say axis, right? So we add the direction arrow here. This is the x-axis according to mathematical representation, and this arrow, this direction towards the up, and this is we call the y-axis, right? So we have now two-dimensional axes, so x and y. And what is the differences between x and y? Apart from the direction, what is the difference? The difference here is that x, unlike x-axis, y-axis has some indicators, right? So the first position here, second position here, third position is here, right? So how we can use that information? Why this is useful? What we can use this indicator for certain information. Let's think about that. In other words, what we cannot do in this x-axis compared to y-axis. So one of the things we can find that is x-axis, we don't know the scales, right? We don't know the data points numbers, right? But y-axis, by integrating these indicators, now we know that this position and this position they have at least twice compared to here, right? So if you compare here and here, you get a sense that at least there are 2 times differences between here and here because you put the indicators, and that means that you can put some scales on here. So let's say this is the kilogram, and this is the 1 kilogram, 2 kilogram, and 3 kilogram. Then you say that this scale is the 1 kilogram units, right? So if you put the scales and units on this axis system, this is add another layers for the arrow, not just the direction, but also for the units as well, right? So, I mean, without thinking about the whole story, we move from here and here. You just look at this graph. This is such a simple graph you see many times during your life. But this graph containing so many components and so many layers, you can describing many different information. So why we have to take the point in this example is that when you draw the plot, you will deliver so many different components you extract from your raw data set, right? So you're not gonna just do the plotting by, I type here and here and make a plot. No, you have to think about the components. You have to sketch the components, and you have to come up with certain ideas how you represent the data set. So this is the critical. This is the thing you have to keep in mind, and you have to keep thinking over and over about your data set. So this is why we're learning the data visualization. Okay, so which components we can think about the data visualization? So the first thing is the data type, right? So which types of the data we're going to use in our analysis. And also you have to think about the data relationship and the plot type, and how, that means that, my data is like this information and what we can represent or what we can empathize from my data set, and which plot type will be the best for representation. Okay, so what will be the colors and alpha? This is another layer, scales and axes, as we see in a direction information, and also text and labels to explain your data set, right? So let's look at some very basic data types, right? So we have at least 4 different data types. So first one is quantitative data. So this represents the different quantity. And the second one is continuous one. There are some information about time, progress, procedures. And there's a data type called discrete. So 4 different distinct entries you want to represent. And the categorical means that there are different the group information within this data set, right? I need to change some colors between here because the categories, this is the 4 different colors, so it's not really distinguished from discrete. So at the point you see this video, I will change the slides for the color. So it has to be red, and gray. Okay. So data relationship and plot type is one of the things you have to keep thinking. There is no rule of thumb. There's no, rule of thumb means that, you press button and get a good result. There is no such things in this, data visualization, and also probably during your life. So what you have to do is you have to keep thinking and finding the relationship in your data set. And we call this as statistics, right? So statistics is describing the observation you made from your experiment or your research. And you keep thinking, then what is the similarities and what is the differences between my observation and the expectation? So getting the relationship of the data set is the key things, and you have to keep doing in the data visualization, and you have to find the optimal way for plotting, right? So when it comes to relationship, most people, familiar with this correlation, because correlation is quite easy things to represent and also thinking, right? So there are some relationship like X and Y. So if you think about the relationship of the data points, you just think about like y equal X. But in a real world data set, the relationship of your data is actually very complicated than you're thinking. So you have to think about the factors or other, underlying differences between the data sets. So finding patterns is not always easy and always not a linear things like you expect, like on Y equal X, right? And also the biological experiment has very unique features about this point. So biological experiments has multiple variables, and also it has multiple different hidden variables. Now we call the covariate, right? So what that means? So easy examples is here. So this is the data set we find from the Geo database, and their description is they're comparing the mice between stressed mice or non-stressed mice, right? So chronic stressed mice is the experimental group, and non-stressed mice is the wild type group, the controls, and they compare these conditions with the different sexes, right? So comparing the males and females. So this looks such a simple. So what will be hidden in this data set, right? So what will be another layers, another dimensions might affecting on this experiment, right? So you have to think about that, because if you go to company, if you go to research lab, institute, thinking about the another possibilities might impact your experiment is critical, right? So you have to keep thinking that what will be another reason to see differences between my experiment, right? So let's think about this situation. We have 4 replicates, so 4 different mice, right? Male and female, stress versus non-stress, stress versus non-stress. So we can guess that there might be some differences coming from assigners. So let's say these 2 mice is processed by me, let's say, and these 2 mice processed by others, right? Somewhat region. We don't know that, right? So all of these mice not processed by same person. So different person, different days, or different places, or different cages. All of that information might make differences in your data set. It might confounding your result. I mean, I a little bit pausing to use the word confounding. Confounding is this word. What? Confounding means that it will make a noise in your data set, right? But you don't know exactly what that is. So that case we call the confounding factors, right? So in a biological research, these things can commonly happen in your data. So you sometimes read about some post or someone's whinging, someone's complaint in a internet, like a break or whatever the biological communities, and they say, biological research is too hard and this is too difficult, and my hand is just a sock and my research is always bad, and I don't know why I'm so cursed, right? The reason is that maybe the person's experiment literally bad. It can be happen, right? But also biological research and biological systems is very complicated. So you don't reckon what is the confounding factors affecting on your data set. So you have to find that confounding factors, or you have to controlling that confounding factors, and you have to minimize that effect in your data set. This is really critical, right? So this is again how we can find that information. This is again visualization. Then you have to find the problems, and you have to find the solutions to think about that origin, right? So this is why we keep talking about the data visualization. Okay, I think I have to explain again by Korean. 제가 원래 녹화를 할 때는 한국말로 다시 설명을 하지는 않는데 요 부분만 다시 한번 설명을 해볼게요. 우리가 데이터 분석을 할 때, 데이터 비주얼라이제이션을 할 때 데이터 릴레이션십과 플롯 타입을 결정하는 게 되게 중요해요. 그래서 내가 지금 보고 있는 데이터가 어떤 관계를 갖고 있는지를 여러분들이 이해를 해야 되는데, 제가 계속적으로 얘기했던 게 이 데이터의 릴레이션십을 보는 과정을 여러분의 하나의 데이터에서 절대 한 번만 보지 않습니다. 계속적으로 여러 가지 방식들을 시도하면서 보게 될 거예요. 그리고 이렇게 보는 비주얼라이제이션의 스텝들이 여러분들이 앞으로 하는, 여러분들이 소위 말해서 코딩을 한다, 프로그래밍을 한다에 거의 70% 이상을 차지하는 부분에 해당할 거라고 저는 생각을 합니다. 그러면 왜 이것을 계속 봐야 되는가에 대해서 여러분들이 이해를 할 필요가 있어요. 뭐냐면 우리가 하는 모든 데이터들은 결국에 어떤 가설이 있고 그 가설을 찾고 그 가설의 의미들에 대해서 생각을 해보는 게 결국에 그 실험 데이터, 생물학 데이터의 가장 중요한 지점이라고 얘기를 할 수 있고, 생물학을 하는 사람들이 가장 매력으로 느끼는 지점이기도 합니다. 그런데 다른 물리나 화학, 뭐 물리도 물론 그렇다고 얘기는 하는데, 소위 말해서 이제 엔지니어링, 그러니까 어떤 프로덕트를 만드는 과정들보다 우리처럼 어떤 현상을 해석하는 그런 학문들은 이 복잡성이 어디서부터 오는지에 대해서 정확하게 알기가 되게 어려울 때가 있습니다. 그렇죠? 우리가 데이터의 관계에 대해서 생각을 할 때 보통 코릴레이션에 대해서 생각을 해요. 그래서 Y는 X다 이런 관계들을 생각을 하는데, 실제로 그거보다 더 복잡한 요소들이 들어갑니다. 그리고 그게 무엇인가에 대해서 생각해 볼 필요가 있어요. 때때로는 생물학 데이터 자체가 여러 가지의 팩터들을 포함하고 있을 때가 있습니다. 그 실험 자체. 근데 또 어떤 경우에는 실험자의 습관이라든지 아니면 여러분들이 실험을 했던 어떤 특정한 상황에 대한 팩터들을 포함할 때가 있어요. 예를 들면 여기서 보는 우리가 이 연구에서 공개된 데이터를 보면 스트레스를 받은 쥐와 그렇지 않은 쥐를 비교를 했죠. 굉장히 간단한 비교죠. 근데 그 비교를 성별에 따라서 비교를 했어요. 그러니까 성별이 암컷인 쥐와 수컷인 쥐, 스트레스를 받은 쥐와 안 받은 쥐 이런 식으로 해서 비교를 했단 말입니다. 근데 여기 보시면 1, 2, 3, 4가 있죠. 이거는 레플리케이트라고 부르는데 우리가 네 번의 반복을 했어요. 그러니까 이 쥐들이 각각 네 마리가 다른 거예요. 왜냐하면 한 번만 측정을 하면 거기서 오차가 나올지 뭘지 모르잖아요. 그래서 네 번을 측정을 합니다. 이것도 약간 여담인데 옛날에는 쥐가 비싸서 한두 마리밖에 못했어요. 근데 요즘에는 이제 쥐도 많이 저렴해지고 실험 기법들도 저렴해져서 이렇게 네 마리도 그냥 무리 없이 하죠. 그러니까 뭐 또 라떼는 얘기지만 라떼는 두 마리 이렇게 해도 많이 했다 이랬는데 요즘엔 점점 커지니까 데이터의 양이 점점 커져요. 근데 어쨌든 네 마리를 했습니다. 근데 이제 이 사람들이 실제로 그렇게 실험을 했다는 건 아니고, 그러니까 제가 가정을 하나 들면 이 두 마리는 제가 실험을 했고 이 3, 4번은 다른 학생이 실험을 했다라고 가정을 해볼게요. 그러면 제가 이 실험을 핸들링을 하는 것과 다른 학생이 이걸 핸들링하는 거에는 당연히 오차가 있을 수밖에 없습니다. 그리고 그 오차가 엄청 심하게 들어올 수도 있고 적게 들어올 수도 있죠. 몰라요. 그 오차라고 하는 건 그때그때마다 다르기 때문에 어쨌든 오차가 발생할 수 있는 어떤 여지가 있다라고 하는 거를 여러분들이 이해를 하셔야 됩니다. 혹은 또 어떤 경우가 있냐면 이 네 마리의 쥐인데 이 두 마리는 점심 먹기 전에 처리를 했고 이 두 마리는 점심 먹고 나서 처리를 했어요. 근데 어떠한 이유에서인지 모르겠지만 점심 먹기 이전에, 그러니까 그 시간의 사이클에 따라서 이 쥐들이 다른 시간 사이클에 대한 어떤 그 반영을 하고 있을 수도 있습니다. 그쵸? 왜냐하면 G라고 하는 게 항상 모두가 다 똑같은 스테이터스에 있는 건 아니기 때문에 시간이나 음식이나 어떤 환경의 노출에 따라서 이 네 마리가 각기 다른 모양새를 갖고 있을 수 있어요. 이 네 마리가 갖고 있는 이 네 마리는 사실 실험 조건상으로는 같죠. 매일이고 스트레스를 안 받은 쥐들이니까. 그런데 그렇게 실험적으로 제어했던 상황임에도 불구하고 전혀 다른 결과들을 가져올 수가 있다라는 거예요. 근데 그것도 마찬가지로 그게 생길 수도 있고 안 생길 수도 있습니다. 그러니까 어떻게 보면 되게 답답할 노릇이죠. 그래서 뭘 해야 되냐? 여러분들이 이 데이터들을 얻고 나서 데이터 비주얼라이제이션을 해보면서 실제로 실험 조건에 따라서 패턴이 나타났는지 아니면 실험 조건 외에 다른 요소들이 개입한 것 같은지에 대해서 여러분들이 충분히 조사하고 데이터 비주얼라이제이션을 할 필요가 있습니다. 우리가 이거를 디텍티브한 작업이라고 불러요. 탐정이 된다라고 얘기를 합니다. 근데 생물학 연구에서는 이 작업들이 굉장히 많이 이루어져요. 그리고 여러분들이 이거를 해야 된다라는 게 기본적으로 여러분들의 머릿속에 있어야 됩니다. 여러분들 뭐 인터넷이나 이런 데 보면 막 생물학 관련된 커뮤니티 이런 데 브릭 같은 데 보면 그런 얘기들 진짜 많아요. 하소연들이. 아, 나는 실험을 정말 열심히 했는데 내 실험 결과는 안 좋고 내 손은 저주받은 것 같다. 막 생물학 실험이 되나 막 이런 얘기를 할 때가 있습니다. 근데 정말로 아이러니하고 정말로 뭐 안타깝다고 얘기하기 좀 그렇지만 정말로 아이러니한 건 뭐냐면 원래 생물학의 현상들은 기계처럼 딱딱딱 나타나는 경우가 없어요. 그냥 없다고 생각하세요. 생물학은 정말 컴플렉스한 시스템이고 컴플리케이티드한 시스템인데 정말 복잡합니다. 그래서 그 복잡성을 여러분들이 포용하지 않으면, 그죠? 그거를 받아들이고 그 복잡성을 최소화하면서 내가 보고 싶은 차원, 디멘전, 제가 디멘전이라는 얘기를 계속 했잖아요. 그거를 여러분들이 끌어낼 수 있는 사람이 결국에 생물학 연구를 잘할 수 있습니다. 그쵸? 그래서 제가 앞서서 계속 생물학 연구도 데이터가 커지고 막 이런다고 얘기를 하는데, 뭐 생물학에서도 맨날 빅데이터 이런 얘기를 하잖아요. 근데 그러면 이제 그런 얘기를 할 때마다 뭐 대학이나 아니면 막 뭐 정부나 이런 데서는 무슨 빅데이터 솔루션을 배운다, 빅데이터 양성 과정을 배운다, 뭐 이런 얘기를 해요. 근데 저는 제가 생각했을 땐 그게 중요한 게 아니에요. 뭐가 중요하냐면 생물학적인 질문을 할 때 그 생물학적인 질문에서 파생되는 데이터가 어떤 복잡성을 갖고 있는지를 보는 생물학적으로 해석하는 능력이 더 필요하다고 생각합니다. 사실상 프로그래밍이나 코딩적인 스킬은 물론 여러분들이 처음 배우기 때문에 어렵지만 그거는 몇 번 하다 보면 여기서 지지플런 연습하고 튜토리얼 하다 보면 몇 가지 비슷한 것들을 계속하게 돼요. 근데 정말로 중요한 건 이 생물학적인 질문에서의 연결되는 복잡성이 어디서 오는지를 논문이 됐건 토론이 됐건 이런 과정을 통해서 배우는 게 훨씬 중요합니다. 그래서 여러분들 중에 회사를 취직하거나 대학원을 가거나 할 때 처음에 학생들이 굉장히 어려워하는 게 뭐냐면 나는 A는 B다라고 배웠는데 계속 새로운 걸 하다 보면 A는 B가 아닌 상황들이 계속적으로 나와요. A가 C가 되기도 하고 A가 D가 되기도 합니다. 그런데 A가 C가 되거나 A가 D가 되는 상황이 어떤 경우에는 분석이 잘못되거나 실험이 잘못돼서 그렇게 나오는 경우도 있어요. 그런데 어떤 경우는 A가 C가 나오거나 A가 D가 나온 게 새로운 발견일 수도 있습니다. 그래서 그게 새로운 발견인지 분석적인 실험적인 오류인지를 분간해낼 수 있는 안목을 가지는 게 그 훈련을 하는 게 훨씬 중요합니다. 왜냐하면 여러분들 회사를 가던 여러분들 대학원을 가던 여러분들은 새로운 지식을 발굴하는 사람이 될 거예요. 정답을 맞추는 사람이 되는 게 절대 아닙니다. 이게 생물학 연구랑 일반적인 공과 그러니까 엔지니어링, 공학이 하는 거랑의 조금의 차이예요. 공학은 물건을 만들기 때문에 A는 B다라고 설정을 하면 A를 넣으면 B가 나오는 상황들을 잘 만드는 게 중요한데 우리는 A라고 하는 게 주어졌을 때 그게 B가 될지 C가 될지 D가 될지 모르는 거예요. 우리는 해석을 하는 사람들이지 우리는 만드는 사람들이 아니거든요. 그렇죠? 그래서 여러분들이 이 데이터 비주얼라이제이션을 배울 때 계속적으로 생각을 해보실 게 내가 주어진 데이터를 어떻게 충분히 해석하고 어떤 플롯의 방식으로 그림을 그려야 될지를 고민을 해보셔야 됩니다. 자, 조금 더 실용적으로 얘기를 하면 우리가 이번 시간에 아마 이 얘기를 하고 끝나겠지만 다음 시간부터 이 여러 가지 플롯의 방식을 배울 거예요. 그러면 처음에 이 지지 플롯 배운 학생들이 잘못된 생각이라고 하긴 좀 그렇지만 어쨌든 좀 그렇게 제가 추천하고 싶지 않은 생각 중에 하나는 뭐냐면 A라고 하는 데이터셋을 여러분들이 받았어요. 그래서 여러분들이 A라고 하는 질문을 하고 싶은데 그 질문에 대해서 하나의 플롯만 그려보고 아, 이렇게 그려야겠다 하고 생각이 끝나요. 네, 그렇게 하시면 안 됩니다. 그쵸? 왜냐하면 여러분들 배우고 여러분들 트레이닝 받는 시간이기 때문에 A라고 하는 질문이 있으면 여러 가지 그래프의 종류들을 다 생각해보고 그려보고 그거에 대해서 해석해보고 뭐가 가장 전달력이 좋고 뭐가 가장 내가 보고 싶었던 디멘전을 잘 드러낼 수 있는지를 여러분들이 계속적으로 생각을 해보셔야 돼요. 그래서 게으르게 하면 안 됩니다. 여러 가지 플로트를 계속적으로 그려봐야 돼요. 어, 이렇게 얘기하면 좀 되게 이상하긴 한데 이게 지금 제가 그 최근에 논문을 쓰고 있는 것 중에 하나인데 이게 지금 한 4년 정도 된 프로젝트예요. 굉장히 좋은 프로젝트가 될 거라고 저는 생각을 하는데 여러분들 이거 한번 보시죠. 이거는 제가 하는 겁니다. 그러니까 교수가 되고 난 저가 계속 코딩을 하는 거예요. 이게 제가 하는 그 코딩인데 보시면 지금 이렇게 보시면 요 하나의 질문에 대해서 플롯을 제가 하나만 그린 게 아니에요. 플롯을 그리고 또 그리고 또 그리고 해서 계속적으로 동일한 걸 여러 다른 방향으로 그려보면서 이게 무슨 의미를 갖고 있고 이게 어떻게 해석을 해야 될지를 계속 생각을 해봅니다. 근데 이게 지금 보시면 뭐 거의 몇 달에 걸쳐서 이렇게 그림들을 그렸잖아요. 그쵸? 근데 이것만 있는 게 아니에요. 아, 이거 제가 지금 여기다가 이거를 꺼놓은 것 같은데, 뉴로 디벨롭먼트 보시다. 왜냐하면 지금 용량이 너무 커가지고 제가 이거를 없애놓은 것 같아요. 데이터. 아, 여기구나. 올드. 자, 올드에 가시면 제가 이 피겨를 당연히 한 번만 그려보지 않고 여기서 여러 번 그렸겠죠. 그죠? 피겨. 2019년 8월에 그린 그림. 아, 지금 이게 업데이트가 되기 때문에 좀 기다려 봅시다. 그렇죠. 여기에 여러 가지 그림을 그렸죠. 그러니까 그림을 그냥 하나만 그려가지고 이렇게 한 게 아니라 여러 가지 다른 아규먼트랑 조건이랑 이런 것들을 넣으면서 계속 그린 거예요, 그림을. 그렇죠. 예, 이게 하나의 폴더. 이게 여기에 한 몇백 장이 있고 여기에 올드에 들어가면 또 몇백 장이 있을 거예요. 지금 이제 싱크를 해야 되니까. 이게 뭐 제가 뭐 학생 때만 그린 거나 그런 게 아니라 지금 연구하는 저도 계속적으로 그리면서 생각을 하는 겁니다. 여러분들 뭐 당연히 지금 이제 배우는 과정이니까 뭐 피겨 하나 그리는 거 되게 어렵고 막 밤새야 되고 이러는데 이게 여러분들 손에 붙으면 붙을수록 한 장 그릴 걸 두 장, 세 장, 네 장 그리면서 계속적으로 생각을 해봐야 됩니다. 그리고 나중에 그 끝은 어디냐? 이렇게 저처럼 뭐 하루에 수백 장, 수천 장씩 그리는 사람이 될 거예요. 그리면서 놓고 해석해보고 아 이게 조금 더 가설을 다듬어야 되겠네 바꿔야겠네 이런 생각을 합니다. 그쵸? 그러면 이거를 언제까지 하냐 이거를 뭐 여러분들 뭐 밤새 코딩하면 저도 뭐 새벽 1시 막 이럴 때까지 맨날 맨날 코딩을 하는데 그러면 이게 나중에 뭐 제가 어느 정도 인생이 편해지면 그때 코딩을 그만하냐 그러지 않겠죠. 제가 연구자로 사는 이상은 계속적으로 코딩을 하겠죠. 지금 이 폴더는 SS 스크래치 스페이스라고 하는데 이거는 제가 하는 그 폴더가 아니라 제 지도 교수님이에요. 그러니까 이분은 벌써 이제 뭐 엄청 그 테뉴어를 받은 교수가 됐는데 저희가 같이 공유해서 작업을 하는 건데 보시면 여기도 이 양반도 그죠? 지금 플롯을 몇 개 그린지 모르겠어요. 계속 그래요, 이 사람도. 그죠? 저도 그리고 이 사람도 그리고 이렇게 그리면 이렇게 그리고 나면 요즘에 그렸던 걸 하나 봅시다. 이렇게 그림을 그리고 나면 생각을 하는 겁니다. 그렇죠? 그래서 이렇게 그리고 나면 저한테도 보내주고 저도 보고 그리고 제 지도 교수인 스테판도 보면서 같이 계속 생각하는 거예요. 이게 무슨 의미를 갖고 있을까? 그리고 내가 의도했던 게 맞을까? 계속적으로 생각을 하는 겁니다. 좀 엄청나게 많은 요 파일들이 여기 이런 식으로 저장이 되어 있어요. 그리고 이런 파일들이 이제 다 끝나고 나면 이제 우리가 최종적으로 전달할 수 있는 하나의 그런 의미에 대해서 우리가 그림을 만들게 됩니다. 그렇죠? 그래서 보시면 이런 식의 이제 그림들이 완성이 되는 거예요. 이제 이게는 논문으로 들어갈 그런 그림에 해당을 하게 되죠. 그죠? 여기도 마찬가지로 이런 식으로 그림을 그리게 됩니다. 이거는 하지만 아직까지 컨피덴셜이기 때문에 딱 끄도록 할게요. 그래서 여러분들이 생각을 하셔야 될 게 우리가 통계를 배우거나 이런 게 그냥 무슨 계산을 하는 거 배우는 거, t-테스트를 배우는 거 당연히 중요하죠. 근데 정말 중요한 포인트는 뭐냐면 내가 실험에서 얻은 데이터, 그리고 누군가가 실험에서 얻은 데이터가 어떤 관계를 갖고 있는가를 여러분들이 답이 정해지지 않은 상황에서 계속적으로 익스플로어를 하는, 탐험을 하는 그 정신을 갖는 게 되게 중요합니다. 이게 말이 되게 이상한데 그 스프릿을 갖는 게 되게 중요해요. 그래서 계속적으로 어떤 관계가 있는지를 해석하고 정리하고 다시 해보고 하는 작업들이 필요합니다. 이 의미를 이해하는 사람들은 굉장히 빠르게 발전을 할 수가 있어요. 그래서 여러분들이 하는 약간 지금 보면 약간 뭔가 약간 노가다 같고 뭔가 약간 막 갈려나가는 것 같은 그런 느낌인데 이게 결국에 여러분들을 성장시키는 여러분들의 안목을 넓힐 수 있는 시간이 된다라는 걸 여러분들이 꼭 기억을 하셨으면 좋겠습니다. 그렇죠? 그리고 여러분들이 더 이런 것들에 익숙해지면 질수록 더 많은 일들을 할 수가 있습니다. 저희 지도 교수가 하는 것처럼 그렇죠? 그래서 이 얘기를 하려고 다시 여기를 했습니다. 그래서 생물학 실험은 그런 컴플렉시티가 있다. 근데 그게 매력 포인트다. 그리고 그거를 이해하기 위해서는 여러 번 여러분들이 충분히 사고할 수 있는 그런 비주얼라이제이션들의 과정들이 필요하다. 요거까지 여러분들이 이해를 하시면 좋겠습니다. Okay, so there was the another, explanation, also some suggestion about why we have to look at the data relationship and plot, and why we need some iterations and many rounds of visualization for your data analysis. And you have to keep that in mind. You're going to find some factors and also relationship from your experiment. And we're going to move on to actual plot information from next time, probably next video, because there is another holiday in 3rd October, right? So I will cover this video as well and upload onto YouTube as well. Okay, thank you very much and see you next time.